Alright, part 3 of chapter 15 focused on treating infections in the body. Here we're looking at antifungal drugs, and with fungal organisms, they are eukaryotic in nature, so they can be very similar to our cells, humans. So the way to target is to target the cell wall. Most fungal cells will have a cell wall. Human cells do not, so we can easily target, to a degree, the cell wall. Also plasma membranes, because there are slight differences, and then nucleic acid synthesis. Now, in terms of the cell membrane, the cell membrane, they are eukaryotic, so there are similarities between fungal cell membranes and humans, but they have a sterol called ergosterol. And this is not found in human cells, but it is actually very similar to cholesterol. So you can use a drug that will target the ergosterol, but you do have to watch for toxicity. Because even though those molecules are different, if there's too much of the drug, it can start to interfere with the cholesterol in our cells and then damage the human cells. Antiprotozoans and anti-helminth drugs target intracellular components, and still these are eukaryotic organisms. They are parasites, and they have very different or complex life cycles. So if you remember with the helminths, generally those organisms will spend at least some time in the GI tract when they infect humans. But these drugs are going to target different stages, and so you may have to be on these drugs long term to make sure you get all of the infecting organisms through that one particular stage where the drug is actually targeting. Now, important to all all infections, you need to be able to identify the susceptibility of an organism with the drug that you want to treat. That gives you confidence that whatever drug you're giving is actually going to be beneficial. So there's a problem with that, though, because organisms are acquiring drug resistance. And what you see here is that the CDC reports that over 2 million people suffer from an antimicrobial resistant infection. So that means you get infected with something and it's drug resistant. The normal drug that might treat E. coli, the normal drug that might treat staph, you can't use because it is resistant. So a very common example is MRSA, methicillin-resistant Staphylococcus aureus. You have to give special antibiotics, different combinations of antibiotics, in order to treat that infection. And unfortunately, approximately 23,000 of those people will die. That's just in the United States. So it's important to determine what the antibiotic susceptibility is to an organism so that you can properly treat it. You don't want to give a person a drug if it's not going to work, because that inappropriately exposes all of the other organisms inside of you to that drug. And so clinical labs do a lot of work with looking at susceptibility tests. Alright, in the next few slides we'll talk about that. There are a couple of different tests. One is going to use agar plates. So you've used agar plates in lab. And there's the Kirby-Bauer test, which is the most common, and then there's the E-test. The Kirby-Bauer test is very, very easy and very cheap, actually, to use. The E-test is a similar test, but it's a little more expensive. And both will determine the basic antimicrobial susceptibility profile for different or for that specific bacterium. And it's relatively inexpensive. Now the E-test is a little more expensive, but we'll talk about that. The Kirby-Bauer test is cheap. So the Kirby-Bauer test is also known as the disc diffusion test. And you spread the bacteria, whatever it is you want to test. If you did it in micro lab, then it's probably just some random broth of E. coli or staph. And you smear it on the complete surface of a Mueller-Hinton agar plate. And you want an equal amount of growth. And then you put discs on top of the surface after you put your bacteria on it. And those discs have a specific drug and a known concentration of the drug. Then you put the plates in the incubator and you look for the zone of inhibition and you measure the diameter. And these are the results. Now you've got your petri dish here and you've got the white growth. If you look at it at the right angle, you can really see the streaks. This would be dipping a cotton swab, sterile cotton swab, into a broth and then spreading it over the entire surface of the plate, turning the plate around, spreading it again, turning the plate around, spreading it some more, so you have just this complete lawn of bacteria growth. Kind of fun. And then you come about and add all of these different discs. Now as you incubate the plate, the bacteria grow, but the antibiotic will move out of the disc and move into, diffuse out into the agar. So that's why it's called the disc diffusion method. And as that bacteria, or as that antibiotic gets released into the agar, if the bacteria is susceptible to it, meaning it's going to die, then you don't have any growth. And so you have this zone of inhibition here. All right, this disc, whatever antibiotic was on there, whatever antibiotic was on this one, didn't inhibit any of the growth. And so you've got bacteria growing all the way up to the disc. The disc, the antibiotic is still moving out, but the bacteria are still growing. So there is, this bacteria is resistant to whatever is on that disc. Now, just because there's a large zone of inhibition that we might see here, and we're looking at the diameter, so that would be 21, 22 millimeters. There are known values, clinical values, tables to compare. And is your sample 21 centimeters? Is it just 15 centimeters? Is it 30 centimeters? Right? You can't base it on just the zone of inhibition here. So this guy has a little tiny zone of inhibition. This guy has a large one. One might naturally say, well, this antibiotic here is more protective than, or more effective than this one. But that's not the case. You actually have to look at each individual antibiotic and compare it to the known tables. So this test will measure susceptibility, will measure resistance, and will measure intermediate levels. And so in any of these cases, you would look for the diameter, you would compare it to the table, and if it's within a particular area, you might say it was resistant, outside of a particular area, sensitive, and then if it's in between the two, then it could be an intermediate. And so then you can utilize that information to determine which is the best antibiotic to actually give the patient. All right, the E-test is very similar, but instead of using discs, it uses strips, and you can identify what's called the minimum inhibitory concentration. This is the drug, or the amount of drug that you can use, the minimum amount of the drug to kill off the bacteria. And so this is the test here. You have this strip, and as you go down or start in either direction, you have an increasing amount or decreasing amount of the drug. So here there is 256 micrograms per mil. That's the concentration of the drug right at this mark. Down here, the concentration is 2. So 2.5, so wasn't quite the correct number or value. But here, at what would appear to be 2.5, all right, because this is the zone of inhibition, so as you increase in the concentration of the drug is going to go out, and you get this teardrop, upside down teardrop effect. And so the bacteria is susceptible to this drug at this point, but once you get down to here, to this 2 microgram per mil concentration, the bacteria is growing up to the strip. So we know that this concentration of the drug is not inhibitory. 0.25 right here, that would be the lowest concentration that inhibits the microbe's growth. Why is that important? Well, in all reality, you want to take as little medicine as possible. All right, and maybe that's because the drug is cheap, or sorry, the drug is expensive, or you just want to take the least amount, so you're taking the least amount of drug. And here there are multiple strips to this one bacteria, and you can see the different size effect, shape effect. All right, this guy here, there's hardly any zone of inhibition, and if this is even resistant, it's probably intermediate at best. All right, so the strips are a little more expensive than the discs, but still fairly practical. And in many cases, you really do want to know what is the minimum amount of antibiotic or drug that you can give a person. All right, another way is to look at broth dilution tests. Right, this determines whether or not a drug is bactericidal, meaning it will kill the drug, or if it's bacteriostatic, meaning it just stops it. And so the antibiotic is diluted, and it's added to a set amount of bacteria, the same amount of bacteria, and you incubate it, and you look for the level of growth measured by turbidity. Right, how cloudy is the broth? Right, and with the broth dilution test, you can also identify the MIC, minimum inhibitory concentration, just like the E test. And then after you allow these broths to grow, then you take a little bit, put it on media that doesn't contain antibiotics, and you look to see if the cells are able to grow. And I don't have a picture of that, sorry. But here, the broth dilution test, so this is a 96 well plate, and there are 12 columns here with 12 different drugs, and increasing amount of concentration. So high amount of drug on this end, low amount on this end. And then how this is actually, how they're identifying growth is by adding this color. And so with no drug in the first row, this is the control. You have ample growth. It's all, you have that nice red spot there. And then you increase the concentration of the drug and look for that red color. Column 9, sorry. If you look at it closely, you've got a little bit of bacteria. Here you might have 2 colonies. Alright, maybe one here, unless those are specs on my computer. But there's a significant decrease. Alright, and then these drugs, alright, MIC not determinable, because this, whatever this bacteria is actually resistant to the drug and is growing in every well. Alright, so this is an easy way to look at what is the easiest, what is the lowest amount of drug you can use. It's actually very fast. So the only thing that changes between any of these wells is what the concentration in the drug is. Alright, and that's where we'll stop for part 3.